My congratulations to Bengt, and thank you to the organizers for the opportunity of speaking about my work today. As the audience is so diverse, I will start with basic information about chromosome structure that you need to know in order to understand my lecture. The nucleosome is the basic unit of DNA coiling in chromosomes. The structure may be described as beads on a string, in which nucleosomes alternate with linker DNA. The existence and main features of the nucleosome were proposed by Roger Kornberg in 1974. The nucleosome contains a set of 8 histone proteins, 2 each of 4 types, termed H2A, H2B, H3, and H4. 147 base pairs of DNA are wrapped in 1.6 turns around the octamer. The 8 histone proteins are arranged as shown here, with the H3, H4 tetramer in the center of the particle and the H2A, H2B dimers towards the periphery, where DNA enters and exits the particle. Double-stranded DNA is rigid, so bending around a particle with a diameter of only about 100 angstroms imparts considerable strain. The energy required for bending is supplied by interactions of negatively charged phosphates in DNA with positively charged amino acids of the histones. This picture was confirmed by X-ray crystal structure determination by Richmond and co-workers in 1997. Here, you see 2 views of the crystal structure. On the left is a view from the top of the particle, with 1.6 turns of the DNA superimposed. And on the right, you see a view from the side of the particle, splitting the particle in 2 between the DNA turns, and viewing from the top, you see 1 turn of the DNA around 4 histones of the octamer. DNA enters the particle from the top left and completes 1 turn at the midpoint of the particle at the top right. The DNA is bound near the end by an H2A, H2B dimer, and in the middle of the particle by the first 2 H3 and H4 molecules of the tetramer. Interactions of the DNA with the histones occur at every point where the DNA strands contact the histone surface, indicated here by the yellow stars. These interactions include charge-charge interactions I mentioned before. All nucleosomes in all organisms share this basic structure. The histone proteins are among the most conserved through evolution. For example, the amino acid sequence of H4 differs by only 2 out of 102 amino acids between plants and animals. The structure of the nucleosome is universal. The nucleosome was long regarded as a packaging device, important for the compaction of DNA in the cell. For example, human DNA, more than 1 meter in length, is compacted more than 100,000-fold during cell division. About 30 years ago, we asked whether the nucleosome also performs a functional role. We investigated the impact of the nucleosome upon gene expression. As most of you know, the RNA polymerase II is responsible for all transcription of a messenger RNA that code for proteins. 2 other polymerases perform more limited roles, transcribing mostly genes for the RNA molecules involved in protein synthesis. RNA polymerase I transcribes genes for the RNA components of the ribosome. RNA polymerase III transcribes genes for the tRNA molecules that bring amino acids to the ribosomes and some small untranslated RNAs. As the first step in the pathway of gene expression, RNA polymerase transcription is a focal point of regulation. We found that the nucleosome performs a critical role in the regulation of RNA polymerase II transcription. We assembled the nucleosome on a promoter consisting of a Tata sequence and transcription start site. The nucleosome prevented the initiation of transcription by RNA polymerase II. This was not a general inhibitory effect of the histones upon RNA polymerase II, because a nucleosome downstream of the start site was no impediment to transcription. RNA polymerase could read right through the nucleosome. Transcription through a nucleosome displaced the histone octamer from the DNA. To summarize, the nucleosome is a general gene repressor. Nucleosomes prevent the initiation of transcription, but allow transcript elongation with the displacement of histones. Soon after this work was published, Hahn and Grunstein showed that nucleosomes interfere with the initiation of transcription in yeast in vivo. They depleted nucleosomes by turning off the expression of the histone H4 gene. Depletion of nucleosomes turned on transcription of a repressed gene, in particular the Fo5 gene, which I will return to later in the lecture. So the nucleosome represses transcription in vitro and in vivo. This leads to the question of how is repression by the nucleosome relieved for transcription. The answer comes from large assemblies of proteins called chromatin remodeling complexes. The first chromatin remodeling complex, referred to as switch SNF, was discovered by Ira Hershkovitz by genetic studies in yeast. When we isolated the complex, we noticed that one subunit had a homologue in yeast. We pursued the homologue and discovered a 1 million molecular weight complex made up of 17 subunits that we termed RISC, for remodels the structure of chromatin. 6 proteins are homologous or identical between switch SNF and RISC, and the corresponding complexes in humans. The human remodeling proteins are mutated in 20% of all cancers, second only to p53, which is mutated in about 50% of all cancers. Switch SNF and RISC differ in both importance and abundance. Whereas switch and SNF genes can be deleted from yeast, RISC genes are essential for cell viability. There are only about 100 molecules of the switch SNF complex per cell, but thousands of RISC complexes. For the rest of the time, I will speak about the essential, abundant RISC complex. Among the functions of RISC subunits listed here, I call your attention especially to the catalytic activity of STH1, the largest subunit of the complex. STH1 catalyzes the movement of DNA in an ATP-dependent manner. We found that RISC has 2 ATP-dependent chromatin remodeling activities. First, RISC can slide a histone octamer along DNA. In the experiment shown here, a nucleosome was formed in the middle of a long DNA molecule and treated with RISC and ATP. Analysis was performed by gel electrophoresis. You see the nucleosome move to the end of the DNA during the reaction. It therefore migrated faster in the gel, forming the lower band after the reaction. Second, RISC can remove a histone octamer from DNA. This chromatin remodeling activity requires both ATP energy and the histone acceptor, which may be another DNA molecule, or, in the experiment shown here, a protein that binds histones, termed a histone chaperone. Products of the reaction are again separated by migration in a gel. The histone octamer is removed piecemeal. In the first few minutes, the H2A, H2B dimers are removed, leaving a complex of the H3, H4 tetramer and DNA. Finally, the tetramer is also removed, resulting in naked DNA. Chromatin remodeling by octamer sliding or removal can expose protein binding sites in the DNA. These may be sites for binding RNA polymerase or sites for proteins that regulate RNA polymerase. Sliding of the octamer exposes sites in the linker region between nucleosomes, while removal of the octamer exposes the entire length of the nucleosomal DNA. Both sliding and removal involve DNA translocation. STH1, the largest subunit of risk, draws DNA into the nucleosome from one side and expels it from the other. DNA associated with the nucleosome, indicated in green, is propelled around the particle and passed into the adjacent linker region. The problem is that DNA cannot easily slide across the histone surface due to the many interactions with histones, symbolized by the yellow stars that I mentioned before. Sliding would require breaking all the DNA histone contacts simultaneously. An idea called Twisty Fusion has been proposed to solve the problem. The idea is that the translocase draws a single DNA base pair into the nucleosome without altering the rest of the structure. This movement involves a twist of 36 degrees, 1/10 of the total, for a full 10 base pair turn of the DNA double helix. The resulting strain is proposed to propagate through the nucleosome, carrying one base pair to the opposite side. The idea could be tested by the introduction of nicks into the nucleosomal DNA, because nicks would prevent the propagation of twisting strain. We found that nicks did not interfere with risk activity, so the idea of twist diffusion was eliminated. We performed structural and biochemical studies that revealed an altogether different solution to the problem of DNA sliding through the nucleosome. In collaboration with Francisco Asturias, we obtained cryo-EM structures of RISC alone and a RISC nucleosome complex. Here, you see in yellow an electron density map of RISC alone at about 25 angstrom resolution. Notice the large central cavity. A difference electron density map between the RISC nucleosome complex and RISC alone is shown in blue mesh. There is little change in RISC structure. Difference density is almost entirely in the central cavity. Rotating almost 90 degrees and removing RISC density for an unobstructed view of the density in the central cavity, there are 3 points of contact with RISC. The histone octamer from the X-ray crystal structure of the nucleosome can be docked into the density in the central cavity. As before, H2A and H2B are red and yellow, and H3 and H4 are green and blue. Here, we remove RISC density from both sides, viewing only a slab in the middle indicated by the dashed box. We rotate again through almost 90 degrees to view from the top down in the lower panel. The X-ray structure of the octamer is shown superimposed on the difference density in blue mesh. One H2A, H2B dimer had to be removed to fit the difference density, and the nucleosomal DNA was mostly undetectable. The dashed line indicates a putative path for DNA detached from the surface of the histone octamer. Detachment of DNA from the histone surface could also be detected by cleavage of the DNA with the enzyme DNase 1. Here, the DNA was radiolabeled at one end, symbolized by the pink star. The fragments resulting from cleavage by DNase were separated on the basis of size by electrophoresis. The column labeled DNA is the pattern produced by digestion of naked DNA. Cleavage occurs more or less at random, with some cleavage sites favored due to the preference of the enzyme for digesting some DNA sequences. The column labeled nucleosome is the pattern produced by digestion of the DNA in the nucleosome. The fragments are multiples of 10 bases. A straightforward explanation is shown in the diagram. Each strand of DNA is maximally exposed to cleavage by the enzyme with the 10 base pair periodicity of the DNA double helix. Between cleavage sites, the DNA is protected by the histone octamer. Binding of RISC to the nucleosome enables attack by DNase at points indicated by the red arrows, halfway in between those seen with the nucleosome alone, indicated by the blue arrows. We interpret this picture as a superposition of two patterns, one of cleavage with the protection by the histone, and the other of cleavage with protection by RISC. The DNA swings from the octamer surface to the RISC surface within the RISC cavity. This is a plausible idea for two reasons. First, we know that the RISC cavity is highly positively charged because we showed that RISC binds a naked DNA molecule with the same affinity as a nucleosome. Second, we know from other studies that nucleosomal DNA is in rapid equilibrium between fully bound and partially unraveled states. DNA is readily released from the nucleosome due to the strain of wrapping on the small radius of curvature of the octamer. Interaction with RISC provides an alternative surface for interaction of the DNA with less bending strain. I have also suggested that RISC may cause a slight alteration of histone octamer conformation. For example, contraction by as little as 1 angstrom, which could help release DNA from the surface of the octamer. The detachment of DNA from the surface of the nucleosome in the RISC cavity solves the DNA sliding problem. Whereas positively charged residues on the histone octamer surface are in register with the DNA phosphates to form the strong interactions needed for bending the DNA, we presume the positively charged surface of the RISC cavity allows interaction in any register and therefore enables sliding. DNA drawn in from one side by the STH1 translocase using the energy of ATP hydrolysis is free to slide to the opposite side. No ATP energy is acquired, only RISC bind energy. Of course, if we add ATP, then the DNA exposed by translocation is cleaved like DNase like a naked DNA molecule as shown on the right. I have also told you that RISC possesses two ATP-dependent remodeling activities: the sliding and removal of nucleosomes. I have also explained that RISC possesses an ATP-independent activity, forming a RISC nucleosome complex in which DNA is released from the surface of the histone octamer. I will now turn to the question of how these activities relieve repression by the nucleosome in yeast cells in vivo. Here you see an example of results obtained by many workers, showing the likelihood of occurrence in a nucleosome as a function of distance along 100,000 base pair region of yeast chromosomal DNA, about 1% of the yeast genome. Below are indicated the locations of RNA transcripts in black and genes in blue in the region shown. A similar picture would be seen for almost any region of any yeast, or for that matter, mammalian chromosome. It is a picture of more or less uniform coverage by nucleosomes, punctuated by occasional absences. Zooming in on the beginning of one RNA transcript, you will see the virtual absence of a nucleosome in front of the gene, a so-called nucleosome-free region, or NFR. Such NFRs are found in front of all genes active in transcription. DNA sequences that must be recognized and bound by gene activator proteins are exposed in the NFR. The TATA sequence, or in many cases a TATA-like sequence important for the initiation of transcription, is also exposed in the NFR. Transcription start sites, however, are generally covered by the first or +1 nucleosome. How is the virtual exclusion of a nucleosome from the NFR achieved? It is widely believed that short stretches of AT-rich DNA in the NFR results in the destabilization and thereby the exclusion of nucleosomes. It is well known that long lengths of the ADT DNA are especially rigid and unable to wrap around the octamer. We have tested whether short stretches of the ADT DNA, as commonly occur in NFRs, can be responsible for the exclusion of the nucleosomes. We inserted stretches of the ADT DNA of 7, 10, and 14 base pairs at various positions from the end of the DNA up to the dyad at the center of the nucleosome. We found at most a twofold difference in affinity for the histone octamer, relative to one of the highest affinity DNA sequences known. Clearly, destabilization by short stretches of the ADT DNA cannot account for the formation of NFRs. What then is responsible? The answer is risk. On the left, you see risk protects the ADT element within a nucleosome from DNA's digestion. On the right, you see a semi-log plot showing the rate of removal of a nucleosome by risk from a natural DNA without the ADT element in red, and the same DNA in which we have inserted a 7 residue, the ADT element, in blue. Insertion results in recognition by risk and removal of the nucleosome. This discovery of a stimulatory effect of the ADT elements explained a surprising finding we made 25 years ago. We found that regulatory sequences that enhance transcription by RNA polymerase I also stimulate transcription by RNA polymerase II. This was unexpected because the control of the two RNA polymerases was believed to be very different. We observed that the RNA polymerase I enhancer contains binding sites for proteins indicated here by blue boxes, and also at the ADT element. We placed each sequence separately in front of a gene transcribed by RNA polymerase II and found an enormous stimulatory effect of the ADT element. We now know the reason that the ADT element stimulated the removal of a promoter nucleosome by risk. Genetic studies in yeast have recently shown that risk is involved in transcription of all RNA polymerases, 1, 2, and 3. Finally, we made measurements of both affinity for the histone octamer and rate of removal of a nucleosome by risk for the ILV1 gene in yeast. We assembled nucleosomes on a series of DNA segments, DNA of the NFR and DNA of each of the first 3 nucleosomes of the gene. As shown in blue, there was virtually no difference in affinity of NFR and gene DNA for the histone octamer. All DNA segments bound DNA almost as well as a high affinity histone DNA binding. By contrast, as shown in red, RISC removed a nucleosome for NFR DNA at least 10 times more rapidly than from any nucleosome containing gene DNA. Now, the experiment was performed on individual nucleosomes assembled in the lab. What might we find for actual yeast chromosomes assembled in yeast cells in vivo? To this end, we isolated a region of a yeast chromosome containing a yeast gene in its native state, in the manner shown here. We inserted recognition sites for a recombinase enzyme on both sides of a gene in yeast, in this case the Fo5 gene, which I mentioned before. We then induced expression of the recombinase enzyme in the yeast, resulting in excision of the gene as a chromatin circle. We isolated the Fo5 circle by an affinity technique. We purified it 200,000-fold to virtual homogeneity. The complete gene circle carried 12 nucleosomes, and because it was isolated in the transcriptionally repressed state, there was no NFR. Rather, the promoter containing the Tata sequence and transcription start site was occupied by 3 nucleosomes. One of these nucleosomes contained the DADT sequence for recognition by RISC, and also a binding site termed UASP2 for an activator protein. A second binding site termed UASP1 for the same activator is always exposed in the adjacent linker region. In other experiments, we placed the recognition site for the recombinase between the promoter and the gene to isolate a promoter circle bearing 3 nucleosomes or a gene circle bearing 9 nucleosomes. We applied RISC and ATP to the chromatin circles and measured the removal of nucleosomes by 2 different methods: topoisomer analysis and limit nuclease digestion. 2 of the 12 nucleosomes on the complete gene plus promoter circle were removed by RISC in vitro, exactly the same number as are removed upon gene activation in yeast in vivo. The nucleosomes removed from the complete circle must have come from the promoter, because none were removed from the gene. We conclude that RISC removes promoter nucleosomes and creates an NFR in an actual yeast chromosome. In further work, we investigated the possible role of histone modification in the targeting of RISC to promoter nucleosomes. Vincent Alfrey discovered in the 60s that histones are acetylated and methylated, and that these modifications correlate with gene activity. More recent work has shown that nucleosomes are also extensively modified by phosphorylation, ubiquitination, and more, mostly on the amino terminal tails of the histones, protruding from the central core of the particle, shown here in various colors. All these modifications correlate with structural and functional states of chromatin, but it is unknown in most cases how these modifications actually affect chromosome structure and function. More than 100 histone modifications have been described. Despite the large number of modifications, they occur in relatively few combinations. In addition to post-translational modifications, there are special forms of histones present at a low level, some of which are listed here. These histone variants are found in nucleosomes in specialized regions of chromosomes, such as the beginning of genes, regulatory regions, centromeres, and sites of DNA damage. Returning to the chromatin circles, it is known that histones of promoter nucleosomes are acetylated. We used the histone deacetylase enzyme to reverse acetylation. We found a marked reduction in the removal of promoter nucleosomes by RISC. At the same time, we observed the removal of nucleosomes from the gene region previously unaffected by RIST. Because of the widespread involvement of RISC in transcription and other DNA transactions, there must be other factors that determine specificity besides the modification of histones. The creation of an NFR is only part of the relief of the repression by the nucleosome. Regulatory and Tata sequences are exposed in the NFR, but not the transcription start site. This point is nicely illustrated by the case of the FO5 gene I just described. Upon activation of the gene, 2 nucleosomes are removed, indicated by dashed ovals, creating an NFR that exposes a regulatory sequence UAS P2. A third nucleosome, the so-called +1 nucleosome, is not removed, but caused to slide, exposing the Tata sequence, but still covering the transcription start site. Transcription is still repressed. Recent work suggests that the detachment of DNA from the histone octamer in the RISC cavity that I mentioned before relieves repression by the +1 nucleosome. Henikoff and co-workers found a partial loss of histone DNA interaction in the +1 nucleosome of active genes in vivo. They showed that association with RISC is responsible for this loss of histone DNA interaction. In conclusion, I told you how we found almost 30 years ago that the nucleosome serves as a general gene repressor. It sets a zero baseline for gene expression. It performs an essential role in cellular processes such as differentiation and development by preventing transcription of all genes except those activated by a specific regulatory protein. 10 years later, we discovered RISC, the most abundant and essential chromatin remodeling complex. RISC relieves repression by the nucleosome. It slides and removes nucleosomes. It is stimulated by the ADT sequences in front of genes, where it creates NFRs, exposing regulatory and Tata sequences. RISC also forms a complex with the nucleosome, in which DNA is released from the histone surface, exposing sequences such as transcription start sites of active genes. These activities of RISC help us understand how repression by the nucleosome is relieved for transcription. RISC also plays roles in replication, recombination, repair, and other chromosomal DNA transactions, doubtless by similar mechanisms. I would like to acknowledge the contributions of my colleagues at Stanford and at the Scripps Research Institute, and thank you all for your attention.
So thank you. And we have time for 1 or 2 questions before we move into coffee.
Can I release this?
So Bengt? Bengt's always ready.
Fantastic talk. You have moved the frontiers. Do you have any comment to Andy Fire's periodic phasing?
Well, we only tried using this sequence, not as a periodic insertion, but per se walking it into the DNA sequence. We have never tried to use it as a repetitive sequence. Okay. We know that, ATs prefer to be at the minor grooves of nucleosomes, not when they come in 4s, when they come in 2s.
If I should be provocative, this is a microscopic phasing. If you look out some horizons bigger, you have the chromosome banding. What's that?
Yeah, I mean, I'm sure that, a lot of this we'll understand much better when we understand higher structures of chromatin. We're at this point just limited to a nucleosome and not beyond. I'm sure many of these modifications and maybe the AT-rich areas, have to do with uncovering much larger areas. So, in 30 years.
So for next big celebration, Bengt, yes. Will be a very big one. Okay, so let's thank the speaker and all the speakers
Thank you.
of this session.